Picture this. It's a warm afternoon, somewhere in ancient East Africa, around 50,000 years ago, and a group has just finished processing a successful hunt. Pieces of meat are laid out on flat rocks near the fire, cooling before the group settles in to eat. The meat is there for approximately 30 seconds before the first fly arrives. Then another. Then approximately 1,700 more, because flies have a remarkable talent for appearing from nowhere the moment food becomes available, as though they've been watching from just outside the frame of reality, waiting for exactly this moment. One ancient woman waves her hand at the situation with the resigned expression of someone who has been doing this her entire life. The flies briefly leave and immediately return because flies did not survive millions of years of evolution by being deterred by hand waving. The meat gets eaten anyway, because in a world where food scarcity is real and the flies' presence has never once actually been linked to any observable change in the food's immediate appearance, it's difficult to develop strong feelings about this particular problem. The flies land, do whatever they do, and leave. The group eats. Most of them are fine. What none of them know, because this information would require microscopes and germ theory to access, is that every single fly that landed on that meat just completed a biological process considerably more complicated and considerably more disturbing than simply touching food and flying away. What actually happened in those brief moments of contact is the subject of this entire video, and by the end of it, you'll either be completely comfortable with flies because you understand the actual risk level, or you'll be considerably less comfortable because you understand what they were actually doing. There is no neutral outcome from this information. Start with what a fly actually is from a biological standpoint, because understanding the insect changes how you interpret everything that follows. Houseflies, the species most commonly landing on human food across most of the world, Musca domestica, are creatures whose entire biology is organized around one primary mission: finding suitable food for laying eggs that will hatch into larvae that need to eat immediately. Everything about their sensory system, their locomotion, their digestive process, and their behavioral repertoire is oriented toward locating organic material, assessing its suitability, feeding rapidly, and potentially using it as an egg deposition site. Flies do not have teeth, which is the first and possibly most significant piece of biological context for understanding what happens when they land on your food. Having no teeth means having no ability to bite off and mechanically process solid food. Flies can only consume liquid or material that has been liquefied, which creates an obvious problem when the attractive food source is solid. Evolution's solution to this problem is one of the more elegant and more unsettling bits of insect biology you'll encounter. Flies carry digestive enzymes in their saliva, which they deposit on solid food surfaces to externally pre-digest whatever they're interested in consuming before absorbing the resulting liquid through their proboscis. The proboscis is the mouthpart flies extend to contact food, a tube-like structure that functions simultaneously as the mechanism for depositing saliva and for absorbing the resulting nutrient solution. When a fly lands on food, it extends its proboscis, deposits enzyme-containing saliva on the food surface, waits briefly for the enzymes to begin breaking down the surface material, and then absorbs the resulting liquid. What appears from the outside like a fly simply touching food and leaving is actually a fly vomiting digestive enzymes onto your food, waiting for those enzymes to work, and then partially consuming the resulting liquefied material. This is the baseline process that occurs every single time a fly makes contact with food of sufficient interest. The enzyme content of fly saliva is functionally significant. These are real digestive enzymes including amylases that break down carbohydrates, proteases that break down proteins, and lipases that break down fats, deployed externally to accomplish outside the body what most organisms accomplish inside it. This external digestion is common across insect groups and represents an evolutionary adaptation that works effectively for flies despite being, from a human perspective, profoundly off-putting when applied to human food. The enzymes themselves aren't the primary concern from a human health standpoint, though they do begin to alter food chemistry at the contact site. The primary concern is what else the fly deposits along with those enzymes. The fly's digestive system is the key to understanding why this matters so much. Flies feed on essentially anything organic, a category that includes food humans find appealing, but also includes decomposing organic material, fecal matter, dead animals, garbage, and any other organic substrate that provides nutrition. This dietary catholicity is excellent for flies, which can find food in an almost unlimited range of environments, but it means the fly's digestive system is a continuous transit environment for the microbial communities present in every surface and material the fly has contacted and consumed. The specific bacteria that flies carry and transfer to food surfaces include some of the most significant human foodborne pathogens known: Salmonella, E. coli, including pathogenic strains, Campylobacter, Staphylococcus aureus, and various others. These bacteria are present in fly bodies because flies have been feeding on fecal matter, decomposing organic material, or other pathogen-rich substrates that are part of the fly's normal feeding repertoire. The bacteria survive within the fly's digestive system, and when the fly deposits saliva on food surfaces, some of this bacterial content is transferred along with the digestive enzymes. The quantity of bacteria transferred per fly contact is variable and context dependent rather than fixed, which is one of the reasons blanket statements about fly contact always being dangerous or always being harmless are both wrong. A fly that has recently fed on fecal matter or decomposing material and then immediately contacts clean food can transfer significant bacterial loads. A fly operating in a relatively clean environment that has only been feeding on clean food sources transfers substantially less. The population of bacteria on any given fly's body and in its gut varies depending on what it's been doing in the hours before contacting your food, which is unfortunately information that is essentially impossible to access through casual observation. The fly's exterior body surface carries bacteria independently of the digestive system, which adds a separate contamination pathway to the enzyme deposition mechanism. Fly legs and body surfaces are covered in tiny hairs and complex surface structures that provide excellent physical attachment points for bacteria from whatever surfaces the fly has walked on. A fly that has walked on fecal material, as flies regularly do because fecal material is a primary food and egg deposition site for flies, carries bacteria on its leg surfaces and body hairs that can be deposited on food surfaces through simple physical contact, even without the proboscis extension and saliva deposition that characterizes active feeding. This is why the standard public health advice about fly contact focuses on the totality of the fly's exterior surface rather than just its mouth. Every surface the fly presents to contact with your food, legs, body, wings is a potential vector for whatever bacteria the fly has accumulated from the environment it's been traveling through. Flies are, in this sense, tiny flying bacteria distribution systems carrying samples from every surface they've walked on and everything they've fed on, deposited on food through the combination of physical contact and active saliva deposition. The fly's reproductive behavior adds yet another dimension to what happens when a fly lands on food, specifically food that the fly assesses as suitable for egg deposition. Female flies lay eggs on organic material that will provide nutrition for hatching larvae, and the assessment of suitability involves sensory evaluation of the food surface through chemoreceptors on the fly's feet and mouth parts. When a fly lands on food and remains for more than a brief moment, it may be conducting this suitability assessment, and if the assessment is positive, the fly may deposit eggs during the same visit that appeared to involve only feeding. Fly eggs are deposited in clusters of 100 to 150 eggs on appropriate substrate, and they hatch into larvae within 8 to 20 hours depending on temperature. Larvae on food represent a contamination outcome that is more visible and more immediately obviously problematic than bacterial transfer, though the bacterial transfer represents the more significant immediate health risk in most cases where humans would actually consume affected food without obvious visual evidence of infestation. The ancient human context for all of this is relevant and important. The ancient woman waving at flies in our opening scenario was dealing with exactly the same biological processes described above, with one critical difference. She was living in an ancient world where the normal human microbiome and immune system were substantially shaped by continuous exposure to the microbial communities associated with environmental contamination, including fly contact. We've discussed this across multiple earlier videos in this series in the context of dirty water consumption and ancient food safety more broadly, and the same principle applies here. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Ancient humans consumed food with fly contact continuously throughout their evolutionary history. The human digestive system, immune function, and gut microbiome that resulted from this continuous exposure were calibrated for managing the pathogen loads associated with ancient food handling environments in ways that provided genuine, if partial, protection against the consequences of what we now recognize as microbial contamination through fly activity. This doesn't mean fly contact was safe or harmless in ancient environments. It means the worst outcomes, serious illness and death from foodborne pathogens, occurred at rates that the population could sustain while still growing, and the individuals most resistant to these pathogen loads had better survival outcomes that influenced population immune function over many generations. The fly-mediated pathogen loads in ancient environments were, in several important ways, different from modern concerns about fly contact with food. Ancient flies operating in ancient environments were accessing fecal material and decomposing organic matter, the same sources modern flies access. But the specific bacterial communities in ancient human environments differed from those in modern environments in ways that affected both the specific pathogens being transferred and the immune calibration of the humans experiencing that transfer. The introduction of new pathogen strains through changes in animal husbandry, agricultural practices, and human population density following the development of civilization substantially changed the pathogen landscape from what ancient hunter-gatherer populations experienced. The behavior of ancient humans around food and flies developed through the same empirical outcome observation and knowledge transmission process that shaped every other aspect of ancient food safety knowledge. Associations between heavily fly-contaminated food, particularly food that had been left accessible in warm conditions for extended periods, and illness outcomes would have been observed across generations, contributing to behavioral patterns around food protection and food freshness assessment that reflected practical understanding of the problem even without mechanistic knowledge of why it existed. Food protection behaviors including covering food with leaves, bark, or other materials, preparing food near smoke that deters flies, consuming food immediately rather than leaving it exposed, and preferentially eating fresh food over material that had been exposed for extended periods all represent behavioral responses to the fly problem that ancient human groups would have developed through exactly this empirical process. These behaviors are visible in various forms across documented traditional cultures worldwide, representing transmitted practical knowledge about protecting food from flies and the illness outcomes associated with heavy fly contact. The smoke dimension is particularly interesting because fire, which ancient humans maintained as a central feature of camp life for multiple purposes we've discussed across this series, also happened to create environments that substantially reduced fly activity. Flies avoid smoke, and camps with well-maintained fires were consequently less hospitable to fly activity around food than camps without fire. This functional benefit of fire maintenance around food was almost certainly observed and incorporated into ancient practical understanding of fire's value, even if it was never explicitly connected to the fly food illness pathway in any conceptual framework. The modern understanding of what happens when a fly lands on food, built from microbiology, parasitology, and epidemiological research, allows us to quantify what ancient humans could only observe qualitatively through outcomes. Studies examining bacterial transfer from fly contact with food surfaces have found that a single fly contact can transfer anywhere from several hundred to several thousand bacteria to a food surface, with transfer rates varying based on the fly's recent activity, the nature of the food surface, the duration of contact, and whether active feeding behavior occurred during the contact. The distinction between brief contact and extended feeding contact matters significantly for risk assessment. A fly that lands briefly on a food surface and departs immediately transfers fewer bacteria than a fly that remains for an extended period of feeding activity. The probability of egg deposition increases with extended contact. The total amount of saliva deposited increases with feeding duration. Brief inadvertent contact while food is uncovered during meal preparation represents substantially lower contamination than food left uncovered for extended periods in fly-active environments where multiple flies make extended feeding contacts repeatedly. The specific foods that present higher risk from fly contact are those that provide warm, moist, protein-rich, or carbohydrate-rich surfaces that both attract flies and support rapid bacterial multiplication following contamination. Meat, fish, and cooked grain-based foods present higher risk than whole fruits with intact skins or dry foods that don't support bacterial growth effectively. The combination of fly contact with temperature conditions that support bacterial multiplication, warm environments rather than refrigeration, creates the highest risk scenarios where a single fly contact can seed a pathogenic bacterial population that multiplies to genuinely dangerous concentrations within hours. This temperature bacterial multiplication interaction is the primary reason that the practical risk of fly contact with food is so much higher in warm conditions than in cool ones, and why ancient humans in warm climates dealing with unrefrigerated food had more immediate reasons to be concerned about fly access to food than populations in cooler climates where bacterial multiplication was naturally slowed. The ancient East African context of our opening scenario represents an environment where the combination of warm temperatures, abundant fly populations, and unrefrigerated meat created exactly the conditions for rapid pathogen multiplication following fly contact, which is precisely why ancient behavioral knowledge about food freshness and protection in warm environments was practically important for health outcomes. The immune system interaction with fly-transferred pathogens is worth addressing directly, because it determines why the outcomes of fly-contaminated food consumption range from no effect to serious illness and occasional fatality. Healthy adult humans with intact immune systems and reasonable baseline gut microbiome diversity manage low-level pathogen exposures from contaminated food through immune surveillance and competitive gut microbiome suppression of incoming pathogens in ways that produce no noticeable illness. The immune system and established gut bacterial communities intercept and neutralize many potential pathogens before they establish meaningful infection. This capacity for managing low-level pathogen exposure is not unlimited, and the threshold between manageable contamination and illness-producing contamination depends on the specific pathogen involved, its virulence, the inoculum size, and the immune status of the individual consuming the contaminated food. Children, elderly individuals, and immunocompromised people face substantially lower safety thresholds than healthy adults, which is reflected in the pattern of severe foodborne illness being disproportionately concentrated in these population groups even when the contamination source affects everyone in a household. The ancient human population immune landscape was in some ways more robust to environmental contamination than modern populations living in highly sanitized environments, because continuous environmental exposure throughout development calibrated immune systems toward the microbial communities present in their environment. But ancient humans also lacked the access to medical care that could address severe foodborne illness when it occurred, making the consequences of the failures of this immune management substantially more serious than equivalent illness in modern medical contexts. The trade-off was better baseline tolerance from continuous exposure combined with higher consequences when that tolerance was exceeded. The fly species that lands on food matters more than most people realize, because different fly species have different feeding preferences and different typical bacterial communities. Houseflies are the primary concern in most inhabited areas because they're specifically adapted to human environments and actively seek out human food and waste. Blowflies and flesh flies are more attracted to meat and decomposing material and carry higher loads of the bacteria specifically associated with those substrates. Fruit flies, which are smaller and more often seen around ripe fruit and fermenting foods, carry different bacterial communities from houseflies and present somewhat different risk profiles. The bright green or metallic blue blowflies that appear at outdoor events carry bacteria specifically associated with decomposing organic matter and represent a higher contamination concern than the smaller houseflies that dominate indoor environments. The 1 minute rule, the popular idea that food dropped on the floor is safe to eat if retrieved within 1 minute, has a fly contact equivalent in the folk wisdom that brief fly contact with food is harmless. Like the floor rule, the fly contact version contains a grain of practical truth in that brief contact transfers less contamination than extended contact. But both rules fail to account for the reality that bacterial transfer is substantially instantaneous, and that even brief contact creates a non-zero contamination event whose significance depends on what the fly was doing immediately before landing. What actually makes the difference between fly-contacted food that's consumed without consequence and fly-contacted food that causes illness is the combination of pathogen load, pathogen virulence, bacterial multiplication opportunity in the food before consumption, and individual immune status. None of these factors are visible or accessible through casual observation, which is why the precautionary approach of protecting food from fly access makes more sense as a behavioral strategy than attempting to assess each specific contact event for its particular risk level. The ancient humans in our opening scenario who ate fly-contacted meat and were mostly fine were benefiting from the combination of practical food freshness, fire smoke deterrents that reduced fly contact to brief events, immune systems calibrated by continuous environmental exposure, and statistical probability that on most days the pathogen loads transferred were within what their immune systems could manage without producing illness. The days when this combination failed, when pathogen loads were high enough, or when a particularly vulnerable individual consumed heavily contaminated food, produced illness outcomes that contributed to the accumulated knowledge about food freshness and protection that ancient groups maintained and transmitted. The full picture of what happens when a fly lands on food is simultaneously more complicated and more nuanced than the simple disgust response it typically produces. The fly is a sophisticated biological system executing a feeding process that involves enzyme deposition, surface chemoreception, potential egg assessment, and bacterial transfer from whatever environmental substrates it has recently contacted, all in the brief moment of contact that looks from the outside like a fly simply touching food. The risk that this creates depends on multiple interacting variables that can't be assessed through observation. The practical response, protecting food from fly access, is the correct behavioral conclusion from this understanding regardless of what any given contact event actually transferred. Understanding what flies are actually doing when they land on food doesn't necessarily make watching it happen more comfortable, but it does replace vague disgust with accurate information, which is a better basis for making decisions about food protection and food safety than aesthetic reaction alone. Ancient humans who developed practical food protection behaviors around fire and covering were making correct behavioral choices for practical reasons. Modern food safety practices that keep flies away from food preparation areas and exposed food are continuing the same rational behavioral tradition with better mechanistic understanding of exactly why it matters. If this kind of investigation into the microbiology and behavioral ecology hiding in ordinary daily interactions with the natural world is your thing, there's considerably more territory worth exploring in this direction. What the microbial communities associated with different insects tell us about the ecological roles those insects play, how ancient human immune function was shaped by continuous exposure to the environmental pathogens that insects transfer, and what the evolutionary relationship between flies and the decomposition ecology they participate in tells us about the role these insects play in nutrient cycling beyond the immediate inconvenience of food contamination. So, stay around, because the biology happening in every ordinary moment of daily life turns out to be considerably more interesting than it appears from the outside.